like to introduce you a little bit on the desert plants and their importance. I will not go much to talk about food security. I will avoid the polemic of climate change, and I will focus mainly on the real science and what can we learn from these plants. But before that, I would like to thank the organizers for this, but also for providing me with this beautiful, oh yeah, can I do it here? No, I will do it here. So actually, with this beautiful bag, which illustrates probably the culture of Saudi Arabia, but for me it was very pleasant to see that it highlights the research that is going on in my lab, which are two things. First is bird proteins. So I'm working on some transcription factors that are called bird proteins. They are involved in asymmetric cell division, and this is including one bird that is called falcon. And so they regulate stem cell divisions, the stem cell niche, growth and tissue patterning, and they form a regulatory network that act in via protein interactions with splicing factors that you can see here, and together they regulate the output, which is the target gene, which is important for tissue patterning. The other pillar of the research is desert plants, and of course mainly the date palms. And date palms are one of the beloved plants to my heart, because and you will see not because of their tastiness, but also because of the amazing future in developmental processes. So deserts have, if you come from Europe and you have a picture of the desert, you see it's a place or a region which is devoid from life. But in reality, this is not the case. So desert has a large amount of life, either animals or plant, and it has a very high vegetative cover. And these plants have really amazing features. They also have been neglected so far. And mainly because we are not aware of their existence, we are not aware of their benefit. So some of them are important, like, for example, the Sodom apple. So this is important. The picture to the left, I will do the traditional way. So this is Sodom apple. It produces fiber and latex, which is very important for pharmacological. Date palm, they have fruits. It's the only plant where you can exploit every bit of it for either for construction, for making carpet, whatsoever. And then we have the extreme habitat plant, which is mangroves, and it's really resistant to salinity, hypoxia, and heat, and also some others with medicinal values. And so growing in the desert so you need to have guts to be there. I mean, personally, I cannot stand 5 minutes in heat at 50 degrees out. So you can imagine what these plants have to cope with. They have heat, which is high temperature. They have high radiation, poor soil, no nutrient, and on top of that, there is grazing, which is a very big problem in addition to the abiotic ones. And we focus and we try to understand how these plants are coping with these environmental conditions and how they are successful in thriving in these habitats. And one major contributor in the plant resilience to this extreme environment is the root system architecture. So this is the roots, and these plants in general are really amazing because they have come with solutions to engineer or design a particular root system for every situation. So if you are a date palm, you will make a very complex roots, as you can see here, with very broad, they make deep roots and very complex structure. If you have a legume, you make deep roots. If you are a grass, you make a shallow ones. And if you are a cactus, you just grow horizontally and a very, very shallow root. But if you are a mangrove, it's even worse because you have to cope with hypoxia and salinity. So what you do, you develop a kind of structure that they call pneumatophores, which allow the plants to get some oxygen, and then that what helps them with the hypoxia. So the way I see it, the roots are like a network. So it's a complex network where you get an input. The input will be the environment. The environment will be like nutrient availability, pathogens, interaction with the microbiome, etc., etc. And the output will be the development. And the development will be portrayed by, for example, either you will have more laterals, which are the side root, either you will have roots that is much thicker or thinner, or either you will have roots that are grow horizontally or laterally. So this is reflected in the anatomy, and I really like these X-ray micro-CT images that really reflect the effect of the environment on root. For instance, here you have a normal root growing very normally, and you can image this in a non-invasive manner. If you deprive part of the root from nutrient, what you can get is one side. The side that is in contact with the nutrient will develop normally. The side that is not receiving any, let's say, any nutrients will not develop. So in the talk, I will focus mainly on 3 different species. They are all living in contrasting habitat. Date palm, which needs fresh waters, it's economically important. Mangrove, they are ecologically important. And Sodom apple, so to date, it has not been studied much. And it's a plant that you can find everywhere in the desert. It doesn't need a single drop of water, and you can see that it's completely green. So the challenging things from studying desert plant is that there is not so much knowledge. So you cannot really do fancy molecular biology, you cannot do genetic study, you cannot do functional biology. But the downside is that whatever you do is new, and you learn a lot of things out of studying them. So we started with the Sodom apple, and you can see they have these deep roots, and then they can form very lateral, nice lateral thinner ones. And once we looked at the germination process, it's amazing because they germinate very fast, they grow very fast, and if you put them in tissue culture, they regenerate much faster than rice or any crop species. And you can see here that during growth, they form this kind of a coat layer, which is formed with root hairs, which is really helping them to attract or to absorb water from the surrounding. So this is the fruit. The fruit is beautiful. It's like an apple, but please don't eat it because it contains latex, and the latex is very toxic. And it's used usually to extract steroid for pharmaceutical reasons. And then you have of course, you have the beautiful flower. The root is very organized, so it's very cylindric. You have different tissue layers. You have the epidermis and the many layers of the cortex, the vascular tissue. You have one layer that is, which you can see here in red color. So these are hydrophobic structures that are important for preventing water loss. And if you look carefully at the root structure, so it's like any normal root, so you have the different nicely organized structure with the stem cells very nicely located in the bottom of the root, and which are surrounding the organizing cells. So the next 2 species are even more amazing because they have started their adaptive strategy much earlier during development. For example, date palm, they use what we call remote germination, where the plant germinates, they make kind of a seed, and then the embryo is dormant within the seed coat. And this is using the cell cycle marker. You can see here that the embryo is completely dormant, while the surrounding, it's grows. It's a multi-layer cellular structure that is protecting the plant from the environmental condition. And when the conditions are right, so the plant will start dividing and differentiated, so then you will form the leaf and the root. The mangroves from on the other side, so the seedlings develop in the tree. So the fruit will open, it will form the seedling, it will make the cotyledon will get transported at the coast, and then once it finds the right support, it will make its anchorage, and it will start making roots. So what about their developmental characteristic and the tissue anatomy? So we looked at the at the root structure, so for both date palm and mangrove, and we can find that both are having, so date palm has a much complex structure consisting of multiple layers of outside layers that are hydrophobic, and we think that this is to prevent the water loss. Then there are these kind of dead cells or irenchyma, and this to allow aeration, and some fiber cells, and the role of fiber cells is to provide steadiness and also to help transporting water. Then the mangroves on the other hand, they are more much simpler structure, and they have also this a much less complex hydrophobic layer surrounding the root. So then we ask, so are these structure, or are these impermeable barriers, or are the hydrophobic barrier, can they reflect the water distribution within the root? So what we did, we measured the water inside the root in a non-invasive manner using the MRI imaging, and where that allows you to look at, to visualize water without disturbing basically the plant. And what you can see here, so if you have high water content, you will have a red color, oranges, and or low will be blue. And you can see that in date palm we have much more water in the outer layer and in the middle, while in the mangrove we have less water in the outside and more around in the middle. So suggesting that in date palm they are very good in keeping water, so they can keep the water much more prominently inside the root, while the mangrove, they do their best to get rid of the water, especially at the outer surface, and they also do their best to get the water out from the vasculature to prevent the vasculature from getting salt inside the plant. So we try to, since these two plants are really good, so they have really they have the potential to have these characteristics or their trait that can be transferred to crops. So we wanted to identify what kind of traits, why are they more tolerant to this condition. So what we did, of course, the usual transcriptomics, and we end up with a list of genes that we have identified. And most interestingly, so we found, as is expected, so there are a lot of genes that are involved in heat tolerance in date palm. Many are involved in hypoxia and osmotic stress response in the case of the mangrove. But interestingly, when we look at the localization within these plants, so using some in situ hybridization, what we found that these transcripts have different localization patterns. They are not ubiquitously expressed, but they are cell type specific. For instance, in date palm, we have this lipid transfer protein, which is a stress response gene. So we found that it's expressed mainly in the vascular tissue and in the. Can you actually see my cursor? No. Sorry about that. So they're in the middle. It's the blue color, while and the mangrove, you can see that it's mainly in the vascular tissue. The peroxidase is mainly in the outermost layer. As you can see here, this cursor is not really working actually. And here. And in the mangrove, you can see the expression of these ribosomal proteins. And for us, if you say ribosomal protein, you expect it everywhere, but no, it really have specific expression only in the vasculature. But if you are looking at stress responses of annual, we cannot restrict ourselves in only focusing either on let's say, roots on transcript, but we also have to look at the metabolic pathway, because these are primary pathways that are affected in stress response. So we did some metabolomic studies, and again, we found very beautiful metabolite, either those that are involved. We identified the pathway that are involved either in salinity, hypoxia, or in heat tolerance. So we are currently dissecting this pathway. But most importantly, so this pathway, they don't say anything, because as developmental biologists, so there is a very nice sentence in the microscopy world, which is seeing is believing. So it's a kind of an MBL logo. So we want to see what we are dealing with. So and to do that, we use the a metabolomic approach, which we call spatial metabolomics, where you can visualize metabolite inside the cells. And there, what we were very happy to see that not only we can look at the spatial distribution of these metabolites, so what you can see, for example, this is a cross-section, longitudinal section of the root, where you can have some metabolites that are mainly in the root cap and the stem cell regions, other only in the transient amplifying cell region, and others that are much lower in the, or more prominent in the differentiation zone. So then we looked at, are the metabolites differentially expressed between the date palm and mangrove, and we found some are very specific for the date palm, so they are present here and not in the mangrove. Others are very specific to the mangrove, not only specific, but they have specific expression patterns, so only in the vasculature and the endodermis. And there are those that are common in both species. So to sum up this part, so what we have learned so far is that these plants have really great potential for adaptation. So they are really prone for adaptation in desert environment. These adaptations start early during the development, so they have already programmed the developmental mechanism in a way that already during germination and already during embryogenesis, they know that they are going to cope with these environmental conditions, so they are prepared for it. They have, so if we can understand how these roots are coping with the desert conditions, if we can isolate these determinants, we can call them either salinity determinant or drought determinant, we can hope to transfer them to crops. We can also engineer different kind of roots. So if you don't want to, if you have a plant or a crop that make deep water, and you are afraid that to deplete the water from aquifer, you can just engineer the root from having a deep root into a shallow root, like the cactus. So there are a lot of engineering to be done in plants, and there is a lot of knowledge that you are gaining now from studying these plants and transferring them into to crops. And there is a big need for combining multiple approach. You cannot use either only genomics or developmental biology or cell biology to understand these processes, but it has to be a combined effort, not from one team, but several teams to move forward with this. So the next couple of minutes I would like to spend is how do we move forward doing some functional biology. And so this is started with the date palm. And why date palm? Because they are very challenging species. So first it's a tree. They grow very, very slow. So in the past, it was said that the one who put the seeds does not live enough to collect the fruit. So that to tell you how long is the period. So it takes something like 10 years to flowers. They have a lot of challenges. They grow very, very slow. They don't regenerate very fast. So what we decided is to create tools, molecular tools, to allow functional genetic in date palm. And to do that, we needed a transformation protocol. So you cannot do genetic, you cannot do any functional biology if you don't have the basic knowledge, which is the transformation. So we started to do, we established this protocol, and this is with the aim or with the objective is to accelerate date palm growth, have it flowering earlier, etc., etc. And the outcome, you will see it in the next slide. So the idea was here is to, from this part, is to prepare colors. So you can take the colors from any plant and then cultivate it and then optimize the pipeline to get plantlet out of it, and then get to the transformation. So the transformation in plant has been very well studied, and lately what we are using, what we call morphogenic regulators. And these morphogenic regulators have been fantastic and very powerful because they allow you to regenerate recalcitrant plants by using stem cell factors. Sorry. Again, using knowledge that has been generated in basic science, for example, using developmental biology. Actually, most of the genes have been isolated from the model plants, which has been ignored for many years by the crop biologists and industry. So thanks to Arabidopsis, we isolated these genes like baby boom, which makes roots, STM mussels, which make shoots, FLC for flower transition. And with this, we could use this. The idea is to use these regulators and to make something that is sorry. So generate something that we called fast palm, and then the idea is to generate a palm that can grow faster by combining molecular biology tools, genome editing technologies, to have them growing in a faster speed than the normal conditions. Okay, so to do this, we did we established the transformation protocols. First, we established the tissue regeneration, and this we established it because for every species, because every species is recalcitrant, so we had to optimize every protocol for every variety. Then we took advantage of this gene, which is Ruby, and it's a visual marker. So to make sure that your genes have been transferred into this plant, so we use this gene, which is reconstructed a whole metabolic pathway, and then when it's reconstructed in plant, you get a red color, and then you can see it here in this primary, in this selected media, and then you propagate it. But of course, the downside of this is that it has a negative impact in plant growth. So then we use the baby boom, and the baby boom is really a morphogenic regulator. It's very important for regeneration and speeding the growth. And there was we improved the transformation from 1% into 12% and then and 18% when we use the wind, which is important for the differentiation. So then now we have kind of a molecular toolbox where we can do transformation, we can do genome editing, we can do crop improvement in this challenging species. And to end up is with this dream, which is what we call farming in the desert. And the idea there, so this is not impossible, so it's already being done in part of the kingdoms where, oh, thank you so much. Sorry, guys. In part of the kingdoms where we can have, so in the desert we have two things. We have the land and we have the sea. So, and if you look at the climate change, so we are losing land, we are losing green, etc., etc. So, and the prediction, for example, in the south of Spain, so I think within 50 to 100 years, it's going to become desert. So how to cope with that? So to cope with that is to start exploiting the desert to generate energy. So we have the sun, so the sun can be used for creating solar panel. We have the sea you can use it for generating desalinated water. You have greenhouses, and you have the palms, so you can use and you have the land, so that's not an issue. So you can combine all these tools and then create what we call the laboratory for the future, where you could design new crops that are engineered to grow in this harsh environment. So to me, I'm not so pessimistic for the future, especially regarding to climate change, as long as we are aware of it, and as long as we are not so melodramatic, but what we only need to find tangible solutions to solve this, the consequence of the climate change. So to end up, I would like to thank people who contributed to the work, especially so Tintin, who is no longer in my lab. She is now full professor in China, so she was working on the date palm and the mangrove. Shasha here, where is Shasha? Yeah, Shasha here, here. So she has been instrumental in setting up the transformation protocol and the pipeline, so she did a tremendous job in a really driving force in the project, and then we have Wouter, who is working on Procera. Of course, the collaborators, Carlo and Salim for the metabolomic and the NMR technologies, Bruker for the MALDI imaging, and of course the funding that is from KAUST. And I would like to thank you for your attention. Apology for the voice and for not thinking that I was pointing, but apparently the pointer was, I was pointing to myself and not to you guys.